has a unique property. This property is known as replication. Replication is the property of the DNA molecule to produce its own identical copy. So capacity to produce its own identical copy. So one molecule of DNA, whenever it is necessary in the cell, can give rise to two molecules of identical DNA. Replication occurs before cell division. Before cell division. So replication of DNA happens every time just prior to cell division. You have studied about cell division. You know that during cell division there is an interface. And in that interface replication of DNA takes place. Suppose there is a diploid cell. This diploid cell, let us say, undergoes mitosis, forms two daughter cells, and these two daughter cells also carry diploid number of chromosomes. That means these daughter cells are exactly identical to the parent cell. The term identical is not just with reference to the chromosome number. It should also be with reference to the amount of DNA. Suppose this cell, let us say, has X amount of DNA, distributed in the 2n number of chromosomes. These two daughter cells also should have, at the end of mitosis, X amount of DNA. X amount of DNA in the parent cell, X amount of DNA in the daughter cells. So if these daughter cells have to carry each X amount of DNA, what should happen to this X amount of DNA? It should become 2x. It should become 2x, so that 1x is distributed to this daughter cell, another x is distributed to this daughter cell. So the two resulting daughter cells will be genetically identical to the parent cell. This is the basic idea of replication. So replication occurs before cell division during the synthetic phase of interphase. The type of replication is described as semi-conservative type. Semi-conservative type. This is because each time DNA replicates, 50% of the parent molecule, 50% of the parent molecule is retained, and the remaining 50%, is newly synthesized. %, of the parental DNA is retained, the other 50% is newly synthesized. Therefore, the type of replication is described as semi-conservative type. It is like this. Suppose this is the DNA molecule. This is the DNA molecule. During replication, the two chains of the DNA molecule separate. The two polynucleotide chains separate due to the dissolution of hydrogen bonds. So, this chain, suppose this is 3' 5' chain, and this is 5' 3' chain. This 3' 5' chain now has to synthesize a new 5' 3' chain. So 50% is parent molecule, 50% is newly synthesized. In this situation, 5' 3' chain is already there. So this has to synthesize a 3' 5' chain. So in the resulting daughter molecules, which are exactly identical to the parent molecule, 50% is the parental molecule, the other 50% is newly synthesized. Replication of DNA is a very accurate process. A very accurate process. Normally, there are no errors. During replication, normally there are no errors. In case there are some errors during replication, there is a mechanism available for correcting the errors. For correcting the errors. Replication always, as we have seen, involves separation of the two polynucleotide chains. Separation of the two polynucleotide chains, and this separation occurs due to the action of an enzyme called DNA helicase. DNA helicase, also called DNA unwindase. Unwinding of the two chains. Unwinding may begin at one place. May begin at one place and proceed further throughout the molecule. This is what we see in prokaryotes. Or unwinding may begin at several places. At several places, this is what we see in eukaryotes. So the point where unwinding begins, the point where replication is initiated, this point is called origin of replication. Origin of replication, and it is abbreviated as ORI. So in prokaryotes, there is only one ORI in the DNA molecule from where unwinding continues. In eukaryotes, there will be several ORI. Unwinding simultaneously begins at several places in the DNA molecule. Let us now see in detail how this replication occurs. Now let us see how replication proceeds. Suppose this is the DNA molecule, and that is the complementary base pairing. This is the 3' 5' chain. This is the 5' 3' chain. Replication always proceeds in the 5' 3' direction. Replication always proceeds in the 5' to 3' direction. As we were discussing earlier, the beginning of replication, the origin of replication, it can be only one, as in prokaryotes, or many, as in eukaryotes. Now let us assume that somewhere here. This is the ORI. That is the ORI. So at this point the two chains start separating, unwinding of the two polynucleotide chains begins at the ORI. So these nitrogen bases are still intact, they have not separated. And here at ORI the nitrogen bases are exposed. At this ORI, a small piece of RNA, is synthesized. At ORI. It is called RNA primer. It is called RNA primer. The synthesis of RNA primer is helped by an enzyme called RNA primase. So a small piece of RNA, is synthesized as complementary to some of the nitrogen bases at the ORI. This is called RNA primer. RNA primer initiates addition of nucleotides to the complementary chain here. So initiates the formation of a new chain. In the 5 prime 3 prime direction. So from here onwards nitrogen bases are added and a new chain is synthesized, and this new chain will be having the complementary nitrogen bases in the 3 prime 5 prime direction, complementary to this 5 prime 3 prime. So from here it proceeds in the 5 prime 3 prime direction continuously as unwinding continues here, more and more nucleotides are added. So in one strand that is 5 prime 3 prime strand, replication proceeds continuously as unwinding continues, formation of the new chain also continues. So this strand is called leading strand. This strand is called leading strand. So this is the leading strand. In the other chain, this is in the 3 prime 5 prime direction. Therefore here addition of complementary nucleotides cannot continue in the same way as in the leading strand. So here short stretches of DNA are formed. This is one stretch 5 prime 3 prime, another stretch 5 prime 3 prime, 5 prime 3 prime, 5 prime 3 prime. Like this, small segments of complementary bases are added. So in the other strand, that is 3 prime 5 prime, replication is discontinuous. It does not occur continuously. Discontinuous. Short segments of DNA are formed, which later these short segments later join to form a complementary chain, So this strand is therefore known as lagging strand. So this becomes the lagging strand. these short pieces of DNA that are formed and then subsequently added, these short pieces of DNA are called Okazaki fragments. Okazaki fragments. So these short segments are called as Okazaki fragments, named after the scientist who discovered and described this phenomenon. So in one strand, formation of the complementary chain proceeds continuously. In the other chain, formation of the complementary chain occurs discontinuously by the joining of Okazaki fragments. So this is the mechanism involved in the replication of DNA. Now here, the enzymes involved in this process are DNA unwindase for separating the two chains, DNA polymerase. It is of three types: DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. Of these three, DNA polymerase 3 is very important because that is the one which joins the nucleotides, which joins the Okazaki fragments together to form a complementary chain of DNA. In addition to this, you have RNA primase and another set of enzymes called tropoisomerases. These are the enzymes that are involved in the process of DNA replication. We have just now seen that DNA replication is of semi-conservative type, wherein 50% of the parent DNA is retained, 50% of the parent DNA is retained, and the other 50% is newly synthesized. This idea was proved experimentally by a team of two scientists, Messelson and Stahel. Messelson and Stahel conducted an experiment in 1958 using the bacterium Escherichia coli. And the results of this experiment proved beyond any doubt that DNA replication is of semi-conservative type. So what they did was, they cultured E. coli Messelson and Stahel cultured E. coli bacteria in a medium containing radioactive ammonium chloride. In a medium containing radioactive ammonium chloride, where the isotope of nitrogen, 15 isotope of nitrogen, was labeled, found that they did it for several generations. Cultured E. coli bacteria in this medium for several generations and found that all the bacteria had their DNA with (15N). This was found to be present in all the bacterial DNA. In the next step, they isolated some bacteria from this culture and cultured them in a medium with 14 NH4Cl. 15 NH4Cl, this is 14 NH4Cl. Removed bacteria some bacteria from this culture periodically and tested them for labeled nitrogen. They found that in the successive generations, here 15 nitrogen 15 isotopes were incorporated in the DNA of all the resulting bacteria. When some of them were placed in nitrogen 14 isotopes NH4Cl, these resulting bacteria in the successive generations found to incorporate 14 nitrogen. So they found that bacteria showed hybrid DNA, that is 15N and 14N. Clearly showing that 50% of the parent DNA and 50% of the newly synthesized DNA will be present in the successive generation of the DNA. The summary of this experiment can be represented like this. Now look at this diagrammatic representation of Michelson and Stahel's experiment. This is the DNA which was found to contain 15 nitrogen, which was repeatedly cultured in 15 NH4Cl for several generations. So nitrogen 15 isotope, they had nitrogen 15 isotopes, heavy DNA. These bacteria were then placed in 14 NH4Cl, N14. You know that most bacterial cells, especially E. coli, divide once in 20 minutes. So after 20 minutes, the sample had double the number of bacteria, and in each of these the DNA was found to contain 14N DNA as well as 15N DNA in equal proportions. So DNA was hybrid, having both 14N DNA, 15N DNA. Another 20 minutes, the next generation. Suppose, assume one cell has divided into two cells, two cells have divided into 4 cells. Now this is the situation. In one set you will have one of this 14N DNA coming from the parent, the other one newly synthesized. So this has both light DNA. Here the other one a complementary heavy DNA, so it will have hybrid DNA. This will have hybrid DNA. This will have again only 14N DNA. So clearly indicating that only 50% is newly synthesized and the other 50% is drawn from the parental DNA. So this experiment, beyond any doubt, proved that replication of DNA is semi-conservative type.